This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.
The Science Notes podcast on OAR FM Dunedin, brought to you by the Division of Sciences at the University of Otago. I'm Dave McMorrin. Over the next half hour, I'll be chatting with a graduate science student about their research, their passion for science, and the things that fuel their creativity. Well, hello, and welcome to Science Notes for another episode. My name is Dave McMorrin, and this time, our guest is Katie Buschang.
Hi.
Hi. Thanks for coming along.
Thanks for having me.
On, I have to say, rather nice Dunedin morning.
Yeah, it's been some good weather recently.
We were in here earlier in the week doing some recording, and it was awful, but today's quite nice, so good.
Not fun.
Good. Which is good for Katie, because Katie's not from here originally. She's from somewhere a bit warmer.
Very much warmer.
Katie's doing a PhD in zoology.
Yep.
2 years in-ish?
Yes, about to have in September my 2-year meeting.
Okay.
So almost the last year.
Yeah, so we're getting through it.
Yeah.
Tell us a bit about how you've ended up in Dunedin.
It's kind of a funny story. So I was finishing up my master's in Florida, and I was just applying and emailing all these professors trying to figure out like where I was going to go. And this one professor kept popping up with all these really cool research articles. And I was like, oh, I want to work with him so bad. And I shot him an email and we started going back and forth. But this was like, I don't know, the 20th professor I had emailed. And so I had stopped doing the deep dive of research into them.
Yep.
And so we were just emailing back and forth. And it wasn't until he was like, okay, I like your CV and your ideas. Let's have a Zoom call and chat. And he was like, all right, let's do Wednesday morning for me. That should be about Tuesday afternoon for you. And I had this panic of, why are you on a whole different day? And that's how I learned he wasn't in the United States or Canada.
You realize it wasn't Dunedin in Florida.
Yeah. I was like, oh, that's a little different. So yeah.
Yeah.
But the Professor Robert Poulin is a fantastic researcher. He truly is astounding in his field, and I feel completely honored to work with him. And I'm so happy to be here and loving New Zealand so much.
I believe he and I got shaved for cancer fundraising together once many years ago. So in Florida, then you did a master's in zoology.
I did a master's in public health, concentration in One Health.
Okay.
So it's kind of it encompasses different fields of public health. So you look at the health of animals, humans, and the environment. And I really liked it because it let me take things like biostats, public health community outreach classes, as well as like hardcore science classes. And I kind of liked that intermixing of the fields. Yeah. And so it was a great master's for me and a wonderful program.
And so then you're kind of coming to your PhD from a slightly different angle than probably a lot of your zoology colleagues are, where they're just all about, it seems like for a lot of the people I've had, and there's just an excuse to spend 3 years being out in the mountains somewhere. But you're coming at more from the health perspective.
Yeah,
right?
And honestly, I for my PhD, I haven't done any field work. So unless you count like a weekend trip to Geraldine, spending a weekend dissecting ducks.
There you go.
In someone's farm shed. That's the most field work I've done, though. So
Geraldine's nice, though.
Yeah, it's a cute little town.
Yeah. So your research then is in parasites.
Yes.
And in particular in toxoplasmosis.
Yeah.
Tell us about toxoplasmosis and why we should care.
Yeah, so Toxoplasma is a disease that, honestly, I hadn't even heard of really until doing my PhD. Like, it was mentioned in a class or two during my master's, but I was like, oh, cool. I think my mom knew more than me, who's a primary school teacher. She was like, oh, yeah, like, if you ever get pregnant, you can't scoop cat poop. You know, you need to be careful around cats, always disinfect the wounds. And, like, that was my knowledge of it. But then when I really got into it and started learning about it during my PhD here, I was like, it's so cool what it can actually do. Like it, according to a New Zealand food safety study from 5 years ago, 43% of New Zealanders are likely infected with this parasite. So congratulations,
so statistically
you have a parasite.
One of us.
Yeah. That number is actually probably lower because that was reported in a New Zealand food safety study 5 years ago. But the actual study was from blood donors 20 years ago.
Right.
And so no one just looks at it because in most people like us, you would present with like a mild cold or flu. But if it gets into like the brain or the eyes, it can cause blindness and encephalitis and lead to death and some of the weirdest ones in very rare cases. But if it gets into the brain, it can cause changes in behavior.
Yeah.
So like you can get anorexia or a shopping addiction or road rage. And so it like changes your whole behavior in this way. And I'm like, oh.
You can blame that on your cat.
Yeah. So, you know.
So what is the thing with the cats then? It says in the notes that you said it's a parasite with a complex life cycle that requires a cat host for the parasite to reproduce. So it needs the cats, right?
Yeah. So without cats, this parasite wouldn't exist. So if you can't reproduce, you can't make offspring, you don't exist. And so the parasite it can only reproduce within a cat's digestive system. And then the cat sheds the cysts in their feces. And then you can pick it up in soil, contaminated water. You know, if something else picks it up and then you eat the tissue of that thing, you can get it. And it's a hardy little parasite. I mean, it can survive for years in soil.
Right. So the cat will poo it out.
Yeah.
And then it will just sit waiting.
For something to pick it up.
Right.
Yeah.
But then when it gets into the other thing, then does it then get out of the other thing?
No.
So how does it,
how
does it get into the cat in the first place?
Usually the cats will share it between themselves or pick it up through the environment from other cats.
Okay.
But if like we got it or a bird or anything else, it kind of just will cause its initial symptoms and then it'll form cysts in your tissue and it just kind of lays dormant there.
Okay.
So unless someone comes and eats your tissue, it's not going to keep transmitting from you.
Cool. So cats are important for the parasite, but they're not so important for you. You're much more concerned about the birds.
Yeah. So a lot of research has already been done on cats, and you kind of know how Toxoplasma and cats are connected. But in New Zealand, no one really tests it for birds. And I did a lit review for my first chapter, and honestly, no one really looked at it in birds. There was a very few studies that I could use from wild birds. It was primarily looked at in like chickens and sheep.
Yeah.
And then in New Zealand, there was only one retrospective study that looked at historic tissues that they had like stored in a sample or in a freezer or something. And then case studies where like a kiwi or like a very iconic bird passed away from a disease like Toxoplasma. Yeah. They tested it and confirmed it was from Toxoplasma. So
there's no research because people didn't know that it was in the birds, or people didn't care?
I honestly am not sure. I think it's more of they don't care because majority of cases are just mild cases and an animal can keep living. But we are seeing like a spike in recent years of increased research and increased concern for the parasite. It's managed to make its way into waterways and is killing like hectares and Maui dolphins. And so we're seeing this spike of research. And I don't know if it's just because it started infecting those or if there's different strains of this parasite. And it could be a different strain has come to New Zealand that's more virulent. And so it's more likely to kill the host than strains previously found in New Zealand.
And sheep. It somehow gets into the sheep.
Yeah, it's really big in sheep. So the only vaccine, the only cure for this parasite is for sheep.
Right.
And it was made here in New Zealand because of how many abortions and birth defects it was causing in sheep. And I mean, it's New Zealand. Yeah. Sheep is the thing here.
Yeah. So when I read that, I thought it's a bit like the TB that gets out of the possums and into the deer.
Could be.
So, well, I mean, not in a parasitology way, but just in terms of the impact on the economy type way. It's a disease that comes from something, and then because it's infecting something that matters in terms of the economy, then we care. Yeah. This is a similar sort of a thing.
Yeah, I would agree with that.
Okay, so birds then. So you're interested then in the prevalence of this parasite in birds.
Yeah.
So so you need to go find some birds, presumably, is the first thing. So how does that part work?
So we are really lucky, or I'm really lucky. I don't have to go find birds. The birds get given to me. So we have a connection that my lab, the Pulin, the parasite lab, has curated through many years, I think early 2000s, late 2000 or early 2010, something like that, is the earliest bird samples. They have this bird pipeline going on where the Dunedin Wildlife Hospital or DOC or a bunch of these agencies, if they get a bird that they have to put down or they found passed away, they will then donate it to our lab and we can test them for, I look for Toxoplasma, but a lot of other people in our lab look at kind of creating this database of overall parasites in these birds so that we can kind of curate. And if something happens to a bird population, we can say, okay, well, these are what we know, this is what's in it, and kind of go from there.
And so does that mean you get lots of different types of birds?
Yes, yeah. So I have seen, I would say, most birds. We don't get kiwis. I just don't think they're in the area.
No.
But also they're a very protected species here.
Yeah.
So they go to more like Massey, like the vet hospital, and they really have a testing for them. But I have seen most birds, I think, in our freezers.
Yeah.
And they hit different. There's definitely like I could dissect seagulls all day, and then we had a penguin come in. And I was just crying over it, trying to dissect this thing, and I was just like, oh, it just hits different. It was like a little blue penguin, and I was just like, oh, and like in a way, it's an interesting thing morally because it's like, well, this bird had already passed away, and without this system, it wouldn't necessarily be going to waste, but kind of like it. Like it wouldn't be putting towards something. And so now because of this, we can put it towards this interesting scientific research that will go back to helping the population. And we also, one of the members of our lab, Jerusha Bennett, I think, set this up where we skin the birds and we donate the pelts to the local iwi where the bird was from. So it's a very good, to me, circle of things of giving back and helping with the research as well.
So you dissect the birds then. Which particular bits of the bird are of interest to you?
So for the second chapter of my PhD, so again, from Lit Review, very little research in this. Everything I could see was they would test different organs in different papers, or they would just blend up the whole bird and test that. So my second chapter was kind of figuring out what would be the best organ to test in birds. So I was taking the liver, lung, heart, brain, and spleen and testing those. And then for the other chapters through the history, because I wasn't here, I've only been here 2 years, they have been taking a piece of the like breast tissue along with a piece of the heart tissue. Right. So going forward into my third chapter, I'll just be testing heart tissues for those. Yeah.
And so then I think from your notes, you're doing DNA analysis.
Yes.
So you take out the heart or the bit of the heart and then you milkshake that up in the way that you do. And then you go and look for bits of DNA that are characteristic of the parasite being there.
So it's through PCR, polymerase chain reaction. And so it's we take it and we digest the tissue and extract the DNA from it. And then I have certain primers that I'll use to test if the parasite is in the tissue.
Yeah, so that's so the primers, it's like a little bit of DNA that you know comes from the parasite.
So it's like a tiny little segment, not tiny, it's like 500 base pairs, but in terms of DNA, tiny. And we know that it's like a repetitive segment, so every single Toxoplasma will have this bit of DNA. And so we put that in as like the marker, and if it has it, it creates this nice little bright band at around 500 on the ladder of the gel.
So, results then.
Results.
What are you finding?
So for the one where we were looking at the different organs, we found it is very much more prominent in the brain and the heart. Right. The brain had a really nice little hot spot, and the heart was the second closest, and then everything else was kind of below it, which is good for me because I only have heart for the third chapter of my PhD.
There you go. So. Is one easier in terms of the dissection?
In terms of the dissection...
Presumably spleens are hard because they're small.
A little bit. Sometimes we do a lab for 200-level students where they help dissect these birds. They used to dissect chickens, and now they dissect these birds, and the samples go to me. And I went and helped out this past year. And one of the main questions I got was, where's the spleen? Is this it? Is this it? I'm like, And sometimes if the bird's been dead a while, the spleen kind of shrivels up and you really can't find it. So
one of these important life skills you get from doing PhDs. You can recognise the spleen in any random bird.
Yep. I didn't expect that to come out of it, but
So you're focusing then on the brain and the heart.
Yeah. And then during that same chapter, we were also comparing different primer sets because, again, from the Lit Review, they weren't matching up. And so we were using two different ones, one called a food primer set and one called a B1 primer set. And the B1 just looks at the B1 gene, the food is called food because they initially used it to look at food products, but it looks at a similar repetitive segment, and we found that the food primers are actually way better at detecting Toxoplasma. They have a lot less false negatives than the B1 do.
So do you do that by testing it against samples that you've deliberately contaminated with toxoplasmosis?
No.
Or?
So we just tested the same birds. So I had 46 mallard ducks, 46 blackback gulls, and 46 red-bill gulls. And I just tested those with the food primers and the B1 primers.
Yep.
Anything that was positive, we sent off for sequencing just at the GAS here on campus.
Okay.
And so we knew that the positives were positives. And so if the food primer picked it up but the B1 didn't, it's a false negative there. So just comparing between the two results gave us our answer.
Okay. So then what amount you had 46 times 3. I can't do that. 138 birds.
Yeah.
Did many of them, all of them, were they infected? Or
it was a good percentage. I would say probably about 30% over species. Some of them were higher. The mallards had 32.6, and then I think the red bills had the highest, and they were 36% or something. Bills. I know.
They've got enough going wrong with them anyway.
So it depends between those three.
Yeah.
What the prevalence was, but about 30% was what I was finding between all three samples.
And is that what you were expecting? I mean, given that, like you say, the international data is patchy, but...
Kind of. Not really. I didn't know what I was expecting. I kind of just jumped in. From the lit review, we found that it is more common in species like owls, harriers, hawks, those like higher predators, and like scavengers. And also ducks. We found in the literature review it was more prevalent in ducks. So ducks we did expect to have a good percentage. But we started testing before I finished the literature review analysis. So it was like, we're finding positives, and then finished it. And I was like, oh, yeah, they have higher prevalence. So probably. But yeah, not really any true.
Is the data in general, when you say there's a high prevalence in owls, for example, is that because of something to do with it being an owl, or is it something to do with how readily you can get the sample? Because I would imagine that black robins, for example, there's probably little data on because there's hardly any black robins, kakapo.
Yeah.
Overseas, there might be birds that are just live in the mountains or somewhere you never see them, and therefore they don't get tested, whereas the ones that you see all the time, like barn owls, then you're more likely to come across a dead one, and then you're more likely to test it.
Yeah. So we did statistical modeling to figure out, what are the factors, and we ran trying to figure out like phylogeny. So that's looking at the evolutionary history. So like, is it more prevalent in barn owls or, a gull?
Yep.
And it took into account like the number of samples. We kind of tried to account for that within the modeling.
Right. Yep.
But there were a lot of birds that just weren't sampled for on our data set. So we can't say for those. And then the data is also probably skewed because no one publishes if you find 0% prevalence. So it's like, well, maybe the bird has been sampled. They just didn't find any, so they didn't publish.
Yep.
So it's, tentative results of like, is it more prevalent? Is it not? But it does give us an idea. And then through that, we found that phylogeny is kind of linked to it. You can visually see in the modeling that we did that there is one, but the statistics said it's not significant. It wasn't like 0.005, the p-value grand standard. But there were factors such as if a bird was a scavenger or an omnivore that made it more likely to have a higher prevalence of Toxoplasma.
Because it's more likely to have eaten something that was itself infected.
Yeah.
Yeah. Than if it just eats seeds or...
Yeah.
Yeah. If your research seems like it's showing that it's actually quite prevalent.
Yeah.
Is there something that we can do about this? I mean, is this an issue does it kill the birds, I suppose, is a question, or is it just something that they live with?
Something for future research.
Yeah.
Depends, honestly, because we have seen, like, through the case studies that I mentioned, it does kill the kiwis and the kia. So you do get these case studies where Toxoplasma did cause the disease, but then it's hard to say what killed a bird in the end because it's like, oh, the bird died from starvation. Well,
Yeah.
Was that starvation just like it was ill from something, it got caught in something? What was the real cause in it? Oh, if it's sick with one thing, its immune system is down, so usually it can catch something else. And then a big debate is with birds, it's Toxoplasma causes blindness. And then they run into a window. Yeah. Well, did the window kill them, or did the Toxoplasma? So it's really hard to say how big of an issue it is, honestly.
Did it die from the Toxoplasma or die with the Toxoplasma?
Yeah. So it's really hard to say. And I think, luckily, that's not a part of my PhD, so I don't have to try to figure that out.
So you have to assess the spread of the issue first before. Yeah. Cool. So you're about 2 years in then?
Yes.
You're busy collecting data?
Yes.
Got a whole bunch of writing up to do. Lots of stats.
Yep, doing that later today.
Excellent. Thoughts for afterwards?
Kind of seeing where it goes. So the job market right now is questionable in all countries I have lived in or been from or come from. And so it's kind of I definitely want to keep looking at parasites and kind of doing this parasite ecology kind of type of research, like figuring out, okay, what influences the parasite? What does the parasite influence? But it's just going to be finding a job that does that. If it's academic, great. I would love to do that. If it's working for something like Doc, also great, as long as I can keep looking at what I want to look at. That's kind of the end goal right now.
Is parasitology an increasingly important thing with climate change?
I would say yes.
Yeah.
So a lot of diseases, so my undergrad and my master's thesis were looking at diseases transmitted by mosquitoes.
Yeah.
And definitely with climate change, we're seeing an increased range of these mosquitoes. And so we're seeing these parasites and these diseases infecting a different range than they used to be able to, and to people that don't necessarily have immune system or are vaccinated for it, like people in tropical areas. So definitely is an increasing field. So lucky to be in it now.
There's in chemistry there's a third year lab course that involves the students being part of something called the initiative. What is it called? DNDI. The Neglected Diseases Initiative. And they're a thing about leishmaniasis, which is a parasite, yes? I think it is, in Africa. And in New Zealand, you don't kind of really know.
I care about it, yeah.
But in other parts of the world, this is huge.
Yeah.
Hugely important.
Yeah.
So, careers in parasitology, it seems to me, should be a thing.
Yeah.
Should be a thing.
Yeah. As long as the funding is there, we'll be good.
Yeah.
It's always the problem, though.
Cool. Well, that's pretty much our time. Thanks, Katie,
for
coming along. All the best for the remaining year.
Yeah,
and
thank you.
The
Hopefully
statistics.
The statistics.
And thank you, everyone out there, for listening. You've been listening to the Science Notes podcast, brought to you by the Division of Sciences at the University of Otago. To listen again, or to find other episodes in this series, go to Otago Access Radio's website, oar.org.nz, or to your favourite podcast platform.
This podcast was produced by OAR FM Dunedin with support from New Zealand On the Air.